Welcome back. So this time we're going to talk about that first step in a little more detail, right? That first step of cellular respiration, and it is called glycolysis and pyruvate processing. All right, let's get started. The first thing I want to talk about is that there are different ways to look at sugar, right? At least conceptually. All right, so there's different ways that you're going to be shown a glucose molecule in this class. So some of these are going to be from your textbook, others of these are going to be from notes, some of these are just going to be from the lecture. So I wanted to address all the different types. So some of these are going to show kind of a stick and right, like little sticks holding together all of these molecules and groups. All right, so that's a ring structure, but it can also be displayed another way, in a linear form, right? With this as being the initial carbon, so carbon 1, 2, 3, 4, 5, and then 6. Okay, so that's another way we can look at it. So down here is a more filling model, right? So this is imagining it more in 3 dimensions, but as you can see, it still has that basic ring structure. And the important thing is that we're looking at a 6-carbon chain that's shaped up like a ring, or can be at least. Even though it's not shaped like a ring here, we're supposed to have kind of interpreted that it is already in that shape. Okay, I hope that helps as we start looking at some of these. Also, I wanted to clarify that for some of the extra tutorial videos that I have posted. They really explain things well, and sometimes, especially when you're dealing with chemistry, you just need animations, you just need pictures. So feel free to look at some of those videos if you start feeling a little lost along the way. All right, really great animations, 3D, it's good stuff. Okay, well, let's get started with our talk. Glycolysis or glycolysis. So glyco is referring to the fact that sugar is sweet, right? So that's helpful. And lysis means to break or to burst open, to shatter apart, okay? So it's lysed. So glycolysis means we're going to break open a glucose molecule, and we're going to get these 2 pyruvic acids. So here, let me draw the second one, because technically there's 2 of them, right? And so what I want you to just kind of imagine is all 6 of those carbons there, right? So let's count it out. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. 6 carbons here. And then we can count these out. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. Oops, still 6. The only difference is we've cut the molecule in half. Glycolysis, glycolysis. Get it? Not too bad. But the splitting of sugar is an ancient process, and it's been pretty good for getting one thing. Well, a couple of things, but not much of anything, really. And that's you get 2 ATPs. Well, really, what happens is you get 4 ATPs, but you have to spend money to make money, and so you have to spend 2 of those ATPs getting those 4 ATPs, so really you only end up with 2 ATPs at the end of the day. The other thing we do get is, remember we talked about those coenzymes, right? So you get one of those coenzymes all recharged up, right? So you get, here's an NAD. So what we do is we're going to add on a hydrogen, and we're going to add on an electron, and we're going to get 2 of these NADH molecules, right? Well, actually, it's like 2 hydrogen and 2 electrons, okay? So hopefully that's helpful. Okay, and the last thing I want to really introduce you to, at least in glycolysis, is the pyruvic acid. All right, so, make it bigger. There we go. We have glucose, 6-carbon molecule, and when we split it in half, it's no longer glucose. Not at all. We've undergone a change, okay? And so that change has given us pyruvic acid. All right, pyruvic acid, or pyruvate. That's another word we use to describe it. So pyruvate, and we get 2 of these, okay? So the thing is we don't have to waste these. Let's break them apart a little bit, or at least move them around and switch some of the components to see if we can get some more ATP, or some more FADA, or NADH, right? So that's what we're going to do. We're going to use this as a tool now to help us get more of those coenzymes that we need, and maybe another ATP or 2. All right, let's look at how. Well, actually, before we look at how, let's look at pyruvate processing. That probably makes more sense. All right, pyruvate processing. So now we're going to take that pyruvate and we're going to change it just a little bit. We're going to modify it ever so slightly, but we're also going to look back at one of these coenzymes, right, that we talked about in the previous video. So here's pyruvate. Hold on, let me get my pen. There's a pen. Okay, so here's our pyruvate, right? 1, 2, 3 carbons. Oh yeah, there it is. That makes sense. So pyruvate is going to get a few things changed. So number one, we're going to lose a carbon dioxide. We're going to pop that right off, okay? We're also going to recharge an NAD to an NADH. Oh yes, we will make another coenzyme. Well, at least complete it, okay? So after that, we're going to pop on a coenzyme, right? That coenzyme is coenzyme A. How easy is that? So here we go. Clipped it on there, and we've capped it. Now it's ready. So this is everything that happens in the cytoplasm, right? Let's talk about a few more things real quick. Okay, number one, what are our scores at this point? Well, we talked about glycolysis. So for glycolysis, we produced 4 ATP. Awesome! But we had to use 2 ATP to get there. Oh, that's kind of a bummer. So we netted out a 2 ATP profit. That's okay. Glycolysis also gave us 2 NADHs. Great, so we have 2 more coenzymes that we can use for transporting protons as well as electrons around in the cell. Those are going to become really important later, so that's nice. But we did not get any FADH2. None of that in glycolysis. Pyruvate processing gave us no ATP. Oh, that's a bummer. But what it did give us is 2 NADHs, so they're charged up and ready to go, ready to transport. But again, no FADH2. That means we're still waiting to see FADH2 come up, right? We're still waiting on that one. So next up is what we're going to do with that modified pyruvate, right? That processed pyruvate. So we're going to enter it into the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle. Okay, that's a heavy hitter. It's not used for ATP very much, although some is produced. What we are going to get out of it are coenzymes, right? That's really the nuts and bolts of what we're going to get out of the Krebs cycle. All right, we'll see you soon.